Okay, welcome to Ask the Expert. Today we have the pleasure of speaking with Dr. Juan Carlos Jaume. He's an MD, he's a professor of medicine, chief of the Division of Endocrinology, Diabetes and Metabolism at the Department of Medicine, and director for the Center for Diabetes and Endocrine Research, CEDER, at the College of Medicine and Life Sciences, University of Toledo, which is formerly the Medical College of Ohio. Dr. Jaume earned his medical degree from the University of Buenos Aires, Argentina, and he completed his internal medicine residency at Albert Einstein, New York. He then completed 2 fellowships, one in endocrinology, diabetes and metabolism, and one in molecular medicine at UCSF. His clinical interest is in thyroid diseases and diabetes, with research interests focused on endocrine autoimmunity and cancer. And he was recently vested as an investigator of the prestigious Helmsley Foundation. So welcome, Dr. Jaume, and we're very interested to hear you talk to us about homing of antigen-specific CAR-T regulatory cells to the human pancreatic islets and preclinical type 1 diabetes reversal. That is very interesting. Welcome, and very interested to hear what you have to say.
Well, thank you for the introduction and this invitation to present my data. Apologize for my voice. I just came off a cold, so bear with me. I'm going to try to be as clear as I can, and most of my slides have the message already already written. So yeah, this is our based the Center for Diabetes and Endocrine Research, CEDER. I show you here our campus, basic science and clinical translational. We perform research, on the topic, and I'm happy to share results. So yeah, a long title. Bear with me. I will go in details of what I'm trying to say here, but homing of antigen-specific CAR-T cells, in this case regulatory cells, to human pancreatic islets, and the results of a preclinical type 1 diabetes reversal trial. So just briefly, and most of the audience may know this, I'm talking about CAR T cell, chimeric antigen receptor T cell. This is a website of a company that produces these cells already for cancer immunotherapy, just to provide an example of how these cells are developed. So as you can see, there is a T cell here, a chimeric antigen receptor construct, and then the product on the right-hand side of a CAR T cell. The process starts by removing T cells from peripheral blood of patients that are in need of the therapy. The second step is taking those T cells into a laboratory and genetically engineer the cells to find a specific target. We call this, giving the GPS address for these cells to find the target is what is done in the lab, and then the cells are returned to the subject for the specific therapy, in this case, immunotherapy of cancer. These CAR T cells have been in the literature for a while, and in the discovery was 1993, and is for an investigator in the research institute in Israel, the Weizmann Research Institute, in which they show how to remove the binding domain of an antibody and hook that up into a T cell receptor. These early CAR T cells were not really clinically relevant, as they did not proliferate, they did not survive for a prolonged period of time. So the investigation continued along the way. Here I show you a timeline of how CAR T cells evolved, and starting with the discovery in 1993. This is the Coley Award that was given for CAR T-cell immunotherapy of cancer to Dr. Michael Sutherland and Dr. Carl Jaume from Memorial Sloan Kettering and UPenn. So the award was basically given when the first publications came about on treatment of patients with cancer with these immune cells. So the first generation of CAR T cells I mentioned were not clinically relevant. However, the second generation, which I highlighted here in 2003, showed that the cells were able to proliferate in vitro by adding a co-stimulatory molecule to the CAR construct. And that's how the science progressed. Within 10 years, the first report on successful treatment came about to 2013. And as early as 2017, they reached the market. Again, CAR T cells for the treatment of cancer. These are cytotoxic immune cells. And these are just the papers that I quoted in 2013 for, in this case, the treatment of acute lymphoblastic leukemia. This is the second paper from Dr. Carl Jaume at UPenn, and it highlights the power of these therapies. Here, 2 children with relapsed and refractory cancer, acute lymphocytic leukemia, were treated with their own immune cells modified as CAR to target a specific protein in the leukemia cells, the CD19, and the therapy worked. So these two patients were able to survive the disease. The cells definitely killed the cancer and proven to be, efficacious. So this was kind of the seminal work that led to the FDA approval of these therapies. Now, just so you can see the power of this technology, these are, we call it the clinical trial universe of CAR T-cell therapies, just pointing out different stages of clinical trials going on, phase 1, phase 2, pivotal here, and only these 2, not 3 CAR T-cells are in the market these days. So lots of research in the background for treatment of, in this case, cancer. This is just to highlight, one of the companies that is generating, several clinical trials against many cancers. Now, this article was provocative in the New York Times some time ago. Can this treatment, that is CAR T cells, that obviously can cure cancer, can it in this case mend the heart? The idea here was to raise the question: can modified immune cells be trained not just to attack tumor, but to do different things? And the picture just show a CAR T cell on attacking on to leukemia cells. So just to give you a graphic view of how the process seems to take place. So it's a cell-to-cell interaction, and there are some cytokines that are being produced by the CAR T cell that end up, in this case, destroying a tumor. So we can definitely say that in the case of CAR T regulatory cells, they can mend pancreatic islets. And what I'm going to show you is our discovery of way to cure autoimmune diabetes in an animal model first, and then try to translate those results into some ex vivo experiments with human tissues. So as far as the immunopathology of type 1 diabetes, chronic autoimmune disease of the pancreas beta cell, which end up eliminated these insulin producing cells as the disease progresses. Here is a schematic representation of a beta cell, the one cell that produces insulin in the pancreas. And I highlight the presence of a target here, the target we use, glutamic acid decarboxylase 65 isoform, a membrane-bound protein that is in charge of converting glutamic acid into gamma aminobutyric acid. So the decarboxylation of glutamic acid renders GABA product. And we know this is a protein present in beta cells, in human and beta cells.
Juan, is this can you that membrane anchorage, is that a GPI linkage?
No, this is a palmitolation domain.
I see.
That is present ubiquitously in GAD65. I can give you more details about GAD65, but basically is a protein present in GABAergic neurons in the brain.
Yes, yes. I know a little bit about it. I think the audience does as well. I just wondered, when you guys are pointing out membrane anchorage,
yes,
what is that? What is that sequence?
So I highlight that point because we know this is a membrane-bound protein, and being a membrane-bound protein is at least on a membrane compartment within the beta cell. The reality is that this GAD65 travels in vesicles, in exovesicles, and so it's not just within the cell, but also outside the cell, at least in the interstitial space that you find in the islets. It's an important feature for what our approach was for these therapeutics. Yes. So I'll make it clearer as we go. So as far as diabetes, we know that effector T cells attack these pancreatic beta cells in an antigen-specific manner, and that glutamic acid decarboxylase antibodies are present in most patients as a primary antibody. As a matter of fact, we use it now commercially for the diagnosis of type 1 diabetes and the definition of which type of diabetes patients have. So again, for diabetes, we don't have a current treatment, and this audience obviously knows that injections of insulin is how we go about. Yes, pancreatic transplants seem to work, but surgery is needed there. So to avoid the autoimmunity against this insulin producing beta cell, cytotoxic T cells need to be downregulated. I mean, that is kind of a given. We know those are the culprits. Here we call them effector cells, TF, and we know that the downregulation of T effector cells or cytotoxic T cells is happening all around this attack by regulatory cells, which are expected to be abundantly present, but their failure obviously tells something different. So if the problem is not enough, or if it's not well fit, if you wish, T regulatory cells to GAD65, then we can explore how to address that issue. And so we thought of treating type 1 diabetes with expanded, patient's own Tregs, modified as a CARB, and in this case specific for GAD65. So our GPS address for the chimeric antigen receptor modification is GAD65 in our case, and I'll show you some of our results next. So again, to avoid the autoimmunity towards beta cells, cytotoxic T cells need to be downregulated, and these regulatory T cells could do that job. So here in the graphic, you can see the modified Treg with the chimeric antigen receptor as we designed. And obviously the idea was that if the cell were to find the antigen, if the cell were to find the target, could deliver this cooling, immune response directly on site. So, regulatory T cells produce different kinds of anti-inflammatory cytokines and also act on a membrane action onto cytotoxic cells. So our goals here were to first develop an animal model of type 1 diabetes that closely resemble the human condition, and we have achieved that, and then designed a GAD65 specific CAR-T therapy with cells that we were able to reach the target in the model and also in humans. We wanted to test that these cells were able to reverse the process of diabetes, and that could translate into the same kind of phenomenon, at least ex vivo, with human GAD65 Tregs and human pancreatic islets. So we, as I said, move on to producing a couple of things, a couple of reagents that were very important for this line of experiments. One was obviously the animal model, which the University of Toledo generated already a patent for. And the other one obviously was the CAR-T cell approach for the treatment of autoimmune diabetes, as well as other autoimmune diseases and transplants. So we have this already on file. And the design of how we did this is shown in here. Basically, we took that antigen, the GAD65 antigen. I show you a schematic representation of an N-terminal domain here, a C-terminal domain, and a middle region of GAD65. This was important for us on the design of which area of GAD65 would we be targeting for these CAR constructs. So we selected human monoclonal antibodies that were able to recognize specific domains of GAD65. These are naturally occurring human monoclonal antibodies that, again, come from patients with type 1 diabetes. We took, obviously, from the T cell receptor, several pieces that are shown in here, the zeta domain, CD28 co-stimulatory molecule, and again, use that monoclonal paratope, that is the recognition domain of the monoclonal antibody, and link it to the chimeric antigen receptor. Again, that was transferred into regulatory T cells. Here is just to show a Western blot of the presence of the CARs on regulatory cells, and also the fact that the transfer of these chimeras onto the T cells was precisely onto regulatory T cells. You can see here the expression of the CD25 and FOXP3, which are the markers of CAR T cells. So as far as the animal model, I give you a hint of how this was done, but it's a work that took years for us to get simplified in this graphic. You can see that we are working in the background of two different mice, C57 Black 6 and the BTBR, we specifically introduced the human GAD65 gene. Mice do not express GAD65, they express GAD67. So if anything, they express trivial amounts of the GAD65 protein. So we needed to have the GAD65 incorporated as a transgene, and we use obviously the human GAD65. And also, in order to make the animals susceptible for the development of diabetes, the human DQ8 MHC class 2 molecule was incorporated. So in this background and with these two transgenes and over generations of impaired fasting glucose, an animal model with spontaneous diabetes, type 1 diabetes was generated. Again, with genetic predisposition, human genetic predisposition, and obviously with the human antigen target of our choice. This is just a scheme of how we went about treating these animals with the CAR Tregs. So I'll show you the details in the next slides of the results, but what you see here is 4 groups of animals, and we work with 2 different chimeras directed to 2 different regions of GAD65. We call them chimera N for N-terminal region, and chimera M for the middle region. These are controls. EPCAM is against just an adhesion molecule, chimera, and also we use a normal Tregs, that is unmodified Tregs as control. So 4 groups of animals with diabetes, all of them very similar, were immunized with these cells, and again follow over a 30-day period. So what we follow was glucose uptake, glucose tolerant test, in which we were able to see that the animals improved over time, and also measure insulin secretion every time we checked for the glucose tolerant test. We also graded the pancreatic islets at the end of the trial, just to see how this intervention modify the profile of immune cell immune cell infiltration of the islets. And again, there is a score being given here for how the profile of T cells changed over that period. So as you can see, for the grading, obviously the animals that were treated with the chimeras that had the GAD65 was obviously much better than what we saw in controls. Let me just show you the next slide. Here, so this is for the glucose tolerance test, the comparison of control group in orange, and also the chimera M treated group, the chimera N treated group, and the group that was treated with plain regulatory T cells, unmodified. And as you can see, at 30 days, and this was really remarkable for what we were expecting here, but at 30 days, there was a clear improvement of the glucose tolerant test on the GAD65 CAR T cells treated groups. We noticed that plain regulatory T cells didn't do much of a job, and this is just the histology of what we found. So on top you have a pancreas of an animal that was not treated, and as you can see, these animals do not have much islets present, and the diabetes, is really shown by the lack of beta cell mass in these individuals. When they are treated with the chimeras, and on the left-hand side you have the chimera M, on the right you have chimera N, within 30 days there is islet regeneration, if you wish. In the case of the chimera M, you still see some islets that are infiltrated, but new islets seem to come about that seem to take over the function of insulin production. And for chimera N, we had a similar situation. This is just to show how the tracking of this GAD65 CAR Treg end up happening in the animal model. So as soon as we infuse these cells, 24 hours after, actually, a sizable amount of cells end up in the pancreas, and also is picked up obviously in peripheral blood and spleen. These are GAD65 CAR T cells that were co-transfected with green fluorescent protein. The pancreatic lymph node, however, lack of all of these cells. So within 24 hours, the cells, yes, reach the pancreas. They are found on the periphery. They're found on the spleen, but they cannot be found on the pancreatic lymph node. And we track these cells in vivo, again, using green fluorescent protein. Here is the result of the summary of the results. But again, you can find them in peripheral blood as well as spleen, and also in the pancreas, but not on the pancreatic lymph nodes 24 hours after infusion. And I hope you can appreciate this. This is 3D disco morphology image of a pancreatic islet. The close-up is basically on this first lane, in red, insulin, and the green fluorescent protein was in the GAD65 CAR T-Rex. Here we also stain for GAD65, which, by the way, co-stains with insulin. And this is just to prove that within 24 hours, the cells are able to find the target in vivo. I think that's kind of what we explain here. When you go from bottom to top, it's just a matter of magnification. But again, the cells reach the islets.
Start eating healthy.
Excuse me?
Sorry, I'm just, I'm admitting someone into the talk. We just had another, someone join us, sorry.
Okay, no, that's fine. So again, the summary of that data for mice is that we immunize the animals, and within 30 days, cells seem to have a beneficial effect. The tracking of the cells showed us that within 24 hours, they reach the islets, and obviously the process of healing start right away. Now, in order to be relevant, this should be shown to, similarly reach human targets. So here I show you basically our in vitro experiments now for human CAR T-Rex. The GAD65 constructs are the same we use for the mice are here being used for modification of human cells. So in these cases, these are patients that were scheduled to have pancreatic surgery for clinical indications, not diabetes, usually pancreatic cancer or pancreatitis. And so what we did is we recruited these patients and first isolated the human T-Rex from peripheral blood. Again, 2 weeks prior to the surgery, the patient donated a sample of blood that we took into the lab, isolate T-Rex, modify them to express the chimeric antigen receptor against GAD65, and then had enough time to grow them and expand them in vitro before the day of the surgery. The day of the surgery, a wedge of pancreas is facilitated to us, and we isolate the islets, as you can see here on the left hand side panel, from normal areas of the pancreas. Again, if this was pancreatic cancer, the tumor site is sent to pathology, and only the wedge of normal pancreatic tissue is the one we work with. So we isolated the islets, and then again co-culture the autologous pancreatic islets with the CAR T-Rex. Again, same individual. Now this CAR T-Reg against GAD65 carrying green fluorescent protein it seemed to find the islet readily, as I said, in the first 24 hours. And here you see the co-staining of GAD65 with the GFP. So I have the specific examples here. We have done this 3 times with 3 different patients. And as you can see, within the 24 hours of co-culture, the camera is already picking up the movement of the CAR-T that recognize the GAD65 on top of the islets. They proliferate significantly. And here I show the 48 and 72 hours, left and right effect on the CAR T cells. So, one of the features of these T regulatory cells modified by CAR is that when the chimeric antigen receptor finds the target, then the signal is for proliferation and cytokine production. So what we can see very clear is that within the 48-hour window, the cells started to duplicate and keep on proliferating on site. This is just the 3rd patient to show that the proliferation continues at 72 hours. And again, at this time, we measure cytokines just to prove that in the supernatant, the dominant species was the CAR Treg production of cytokine. So try to go fast on this. Hopefully we have time for questions, but
Sure, yeah, we definitely do. We can continue a little bit longer.
Yeah, so the summary is basically that for us it's the first time to observe a therapeutic approach that was successful in abrogating the diabetes in our mouse model. We have tried many things in this animal model, and as opposed to NOD mouse, these animals do not get better with interventions. So this was the first time we were able to revert the phenotype of diabetes on the 30-day trial. I just wanted to emphasize that. Also, the antigen-specific proliferative capacity of these GAD65 CAR constructs was 5 times higher than that of naive normal Tregs, and this provides evidence for superiority of these CAR Tregs over normal Tregs on diabetes and reversal. Finally, we are moving on to human experimentation in vitro, of course, with this homing of engineered GAD65 CAR Tregs onto human autologous pancreatic islets. And this proved to us that the same contract that were used before for the reversal of diabetes in mice are also potentially useful in human clinical trials. So I have one more slide for future directions. Obviously that the next step for us is to move this into a human trial, a phase 1/2A clinical trial. The scheme is basically very, very simple here. Is taking peripheral blood from patients. We know that we can find these regulatory T cells in enough volumes, in enough amounts, just with 2 teaspoons of blood. So 10 cc's of blood is what we have been using, and provides enough regulatory T cells for us to do the chimeric antigen receptor modification in vitro and the expansion. So hopefully, as I said, we can translate this into clinical trials in the near future.
Thank you very much. This is a fascinating approach. I applaud you for taking the lessons from cancer therapy and bringing it to type 1 diabetes. I had a couple of questions. It says here you guys are going to begin using collected PBMCs from LADA patients. Now, why did you choose LADA patients versus just the endotype maybe in a 2-year-old or a 13-year-old, a purely type 1 diabetes patient?
No, so what I wrote there is type 1D dash LADA. So we will include both.
Okay, great. That's wonderful.
The idea here was, if we were to choose a population, we would like to start with, early onset diabetics or new onset diabetics, just because of the impact that this could have on them. But potentially, we think that, this approach may be suitable even for patients that have had diabetes for many years.
Can
So-
I ask you, in an earlier, though, that's great. In an earlier slide, you showed that the patient would have to have a short course of chemotherapy prior to the infusion. Would this be the case here as well?
No, for cancer, that has been the approach, that is only use the CAR T cells as a second line of therapy.
Right.
For diabetes, that will not be the case whatsoever. So the idea here is to replenish regulatory cells that are specific for the target, and there is no need for any kind of intervention prior to the CAR T-Reg treatment.
That's also great. So unlike, some of the other islet implantation approaches, there will be no need for immune suppression.
Correct.
And then I also want to ask you with the, what's the thought around, the TMEMs, the memory cells? Because those are right. Once the patient has type 1, the memory cells are, kind of hanging out in the lymph nodes, ready for some other insult to happen where they, they're reactivated. So what about those? Would this be, so you'd have this treatment with the infusion of the CAR T cells. What if later down the line, the person gets, Coxsackie or a viral insult, and then of course the islet, is under more stress and perhaps elicits another immune attack from the memory cells? Would that be a cause for another infusion, or what are you thinking?
So yeah, this is the beauty of this CAR T cell therapy. We know now that the CAR T cells stay with the individual for a decade. So, this comes from the cancer treatments. So we know that the CAR T cells go and do their job up until, as the antigen is available to them. So in the case of patients with leukemia, when the leukemia comes back, there is a resurgence of the CAR T cells that were infused in the first place. So there is no need for retreatment whatsoever. The case of diabetes, we think is going to behave the same way. That is, the CAR T regs will be present there. And as soon as there is a need for the presence of regulation, these CAR T cells always have that antigen available to them. The more antigen that becomes available, that is the potential destruction of new beta cells, will bring about another wave of Treg expansion. And so kind of, controlling the situation on their own for the life of the individual. Again, this is hypothetical. We have not done the studies for more than, as you can see, 30 days. But the hypothesis here is that these CAR T proliferate on demand. Whenever the antigen is available, they proliferate. And since they are at the site, the cooling effect of the immune response will happen at the same time the attack is happening.
Yes. Okay, that's interesting. So clonal expansion on demand.
Mm-hmm.
And what about, oh, sorry, I want to extend this to people in the audience. Would anyone like to ask a question? Please feel free to unmute yourself or raise your hand. I see several people are there, but no one's raising their hand. I guess my final question is, how do you see this technology sort of dovetailing with what Sonoma Bio in the Bay Area, headed by Jeff Bluestone, is trying to accomplish? Or, do you guys have a collaboration? Are you running after the same technology? How do you see the technologies, the two technologies, sort of moving the ball forward?
So we do not have specific, as you know, there are several companies now, not just Sonoma Therapeutic, but Avatar in the East Coast, you know,
Yes.
Mathis and even, you know, in Europe. So the approaches have been different in general. For what we know, we were the first ones coming up with this, you know, and that's why we have this technology patent. I don't know what Sonoma, you know, is looking at. I know they're working on rheumatoid arthritis and a similar approach for rheumatoid arthritis. And you know, as I said, Avatar is concentrated on MS, multiple sclerosis approach. So I think, you know, I mean, we are open to collaborations, of course, and we would, as I said, like to move this into a clinical phase as fast as we can. We need all the help we can get for that to happen. So, so yes, you know, I think, I mean, it's comforting to see that, you know, this big, you know, I mean, personalities in the field are moving forward with this technology as well. So we are really, you know, I mean, honored to be part of the of this wave, and yeah, would like to see happening soon.
Yes, it would be great to get, you know, on a fast track with the FDA and get the trials moving. I think that the, you know, the support by Helmsley is a vote of confidence that you guys are on the right track and that, you know, potentially you can move this forward quickly. And I would encourage anyone who is out there who's listening that has an interest to reach out to Juan, to Dr. Jaume and speak with him directly about potential collaborations or, you know, other things that might move this forward quickly.
Well, thank you for that pitch.
Thank you, Dr. Jaume. Thank you again for speaking with us today. And we wish you all the best for moving this technology forward.
Thank you. Thank you for this opportunity.
Have a great day. Bye-bye.
Bye-bye.